Hi everyone, I'm Dr. Fatma Kaplan, CEO and founder of Heroísmo. I would like to thank organizer and particularly Anna Presh for inviting me to share our project Nematodes in Space. Insect-killing nematodes are microscopic roundworms, and they're also known as entomopathogenic nematodes, EPN, or beneficial nematodes. They are used actually as a model organism to study biology of parasites. They're also important commercially to control insect pests for providing eco-friendly solutions. The picture here is a consumed insect host, and the rope-like structures are a whole bunch of beneficial nematodes leaving consumed insect. Entomopathogenic nematodes enter the insects from natural opening, mouths or the cuticle, and once they're inside the insect hemolymph or insect blood, they release their symbiotic bacteria, which kills the insects, feeds on the insects. Nematodes feed on the bacteria. Once the insect is consumed, nematodes leave the insect is mass exodus. Even though this picture shows that these nematodes, they're called infective juveniles or IJs, find this healthy host really fast with that arrow, the reality is a lot more complex. There are many signals involved in these nematodes to find a new host. One of those signals I found is a pheromone mixture. Pheromones are signaling compounds that are released from one member and affective behavior and physiology of the other members of the same species. We are interested in pheromones because they can be used commercially and improve the effectiveness of these nematodes. In nematodes, these pheromones are called ascoricides. It's a class of compounds, and the behavior we're interested is dispersal behavior. In the absence of pheromones, they stay put, they stay in one location. Once we add the pheromones, they disperse. Now they're dispersing and searching for a healthy insect. In this case, it's agricultural pest. So when we had the opportunity to send the nematodes in International Space Station, the first question we had, how was the pheromone production affected by microgravity? Then as we were thinking about how to design this experiment, we realized we had a lot of things we didn't really know, even though the answer to these questions are already known on Earth, but not in space. The first question we had, well, in microgravity, can they actually leave the consumed insect host? And then, can they actually move to the soil? Because they do sinusoidal movements. On Earth, the soil is held by gravity. They have capillaries they go through. And then their target insect is also held by gravity, so they can attach and go inside. What about in space? It's floating, and the nematodes are floating, and we didn't really know actually they could directionally move, because in water they cannot. And then what about the insect immune system? Would it be really vigorous and kill the nematodes? Let's say everything went right, and then can they develop in space and reproduce? And even though this may be simple, just one factor we changed the microgravity from gravity to microgravity, but microgravity affects a lot of things. For example, water. Nematodes live in the water interstitial spaces in the soil. Water behaves very differently in microgravity versus on Earth. These nematodes are very sensitive to moisture and the soil type and dehydration. And microgravity affects other things like gravitational loading, hydrostatic pressure, and not to mention they have to cooperate, two organisms, it's symbiotic bacteria and the nematodes together to infect a multi-step infection process. So it is difficult to predict the outcome of this experiment. And we had only one chance. Once we send the experiments to International Space Station, we cannot change anything. So we had to think about all these steps, whether they could emerge, go through the sand, find the insect, overcome its immune system, and reproduce in space. So we had 4 different samples for this one. We also had extra controls. We had the same set of experiment we run concurrently with space. This experiment was also time sensitive, so we had to prepare 2 days before the flight at Kennedy Space Center, and we were lucky we had a really communicative implementation partners, and they were able to get the nematodes on the shuttle on December 5, actually a little earlier. And on December 5, once the shuttle is in space, it was just 10 minutes. Then the microgravity experiment started. And January 7, it returned a month later. This is me unpacking our samples in California. We had another set of samples that went to our collaborator, Byron, Georgia. They were also unpacking at the same time. So it was a success. They were able to emerge in space, went through 10 centimeter soil, moist, infected the insect, and reproduce. We were very happy, but we also had an unexpected result. The nematodes, the ones right here emerged, could not survive when they come back to Earth. And we had a lot of control, so we immediately looked at corresponding Earth control because we thought maybe they ran out of air and just died. And we looked at the Earth control. They were happy and moving around. So we thought, well, maybe they didn't run out of air. But the problem is microgravity is a very dynamic environment. And the oxygen and the carbon dioxide is distributed differently. So the Earth control didn't quite answer our question, the space-induced hypoxia, which is a very well-known phenomena. Now, I want you to remember we have 2 infections in this experiment. In one case, where the infection occurs in space, and the baby nematodes here, the new generation, never experienced gravity during their development. And we had another first infection where we did the infection on Earth, they emerged in space. These nematodes, during their life as an egg or early juvenile, they experienced gravity even though they were born in space. So here is our second infection nematodes. They were bent, literally bent, and they were dead. And then we looked at our other experiments, whether those actually died, if that was the space-induced hypoxia. And from the first experiment, these nematodes were moving sinusoidal, and they were happy, so this was not a space-induced hypoxia. It suggested that these nematodes, if they never experienced during development microgravity, they could not adjust to Earth's microgravity. Now, one other question you might be thinking, what if they're just stayed in the space? They could be. So we also had grandparents of these nematodes. We sent them in another control. They stayed in space for a month, and they also experienced microgravity before they went there, and those nematodes were also fine, just like these nematodes. So, in summary, nematodes can emerge in space, they can infect in space, so we can actually have successful biocontrol if we were to colonize space and the moon, and if we were to have agriculture. Without agriculture, we cannot colonize space. I would like to thank our collaborator, Dr. Shapiro Elon, and our incubators, accelerators, and our funding sources providing us funds and infrastructure. So this is a one tiny step for a nematode. It is a big step towards sustainable agriculture. So we published this work in 2020 in one of the Nature partner journals, MPJ Microgravity. Thank you.